Hey there, and welcome to This Explainer. Today, we are diving into an incredibly fascinating, completely unseen world that dictates almost everything about how our planet and our own bodies function. We're talking about the microscopic universe. Microbiology is the specialized area of biology dealing with living things that are just way too small to be seen without magnification. And as we're going to see today, these tiny, invisible entities, they are responsible for some of the biggest impacts on Earth. Let me start off by asking you a question. We constantly see advertisements telling us that bacteria and viruses are lurking all over our homes, right? And that we absolutely need to buy harsh antibacterial cleaning products to eradicate every last one of them. But should you? Is a totally sterile environment actually the healthiest one? Well, let's dive into this and see why that urge to destroy all microbes might actually be entirely backward. By the end of this explainer, I promise you, it might just view the quote-unquote germs in your house in a completely different light. Here is our roadmap for today's explainer. First, we'll meet the invisible world. Then, we'll look at how we actually name them. After that, we'll explore how they shape our planet, solve a few microbial mysteries, and finally, dive into the modern microbial revolution. Section 1. Meet the invisible world, focusing on the sheer scale and diversity of microbes. Okay, before we meet the actual roster of microbes, we really need to understand the fundamental divide in this microscopic universe. When we look at cellular architecture, we basically see two distinct types. First up, prokaryotic cells. These are simple, single-celled organisms that completely lack a membrane-bound true nucleus to hold their DNA. Their DNA is just circular and floating freely inside. Contrast that with eukaryotic cells. Eukaryotic literally translates to true nucleus. These cells contain a specific, protected, membrane-bound nucleus that holds their linear DNA. Basically, every living thing you can see with the naked eye, plants, animals, fungi, is made up of these more complex eukaryotic cells. So who exactly is on this microbial roster? Well, we have bacteria and archaea, which are both single-celled prokaryotes, though archaea tend to be extremophiles. They love incredibly hot or salty environments. Then we have our microscopic eukaryotes, fungi, like yeasts and molds, protozoa, which are single-celled and kind of animal-like, and algae, which are photosynthetic. We also study helminthes. Now, these are multicellular worms, and while an adult tapeworm is definitely not microscopic, they have microscopic stages that cause disease, so they make the team. And finally, we have viruses. Now, viruses are wild because they completely break all the rules. Biology always has an exception, and viruses are a massive one. They are acellular, meaning they aren't made of cells at all. Because cells are the fundamental unit of life, viruses are actually considered non-living. They're what we call obligate intracellular parasites. They absolutely must get inside a living host cell to reproduce, simply because they don't have their own cellular machinery. Structurally, they are incredibly simple. It's pretty much just a protein coat surrounding some genetic information. And uniquely, they carry either DNA or RNA, but rarely both. Section 2. How we name them. Let's navigate the massive, chaotic tree of life. Scientists desperately need a way to organize this unbelievable diversity. So we use a hierarchical classification system that starts super broad and gets incredibly specific. A really great way to remember the order of this hierarchy is a simple mnemonic: domain, kingdom, phylum, class, order, family, genus, species. You can remember this by thinking, do keep pots clean or family gets sick. It goes from the widest umbrella, the domain, all the way down to a very specific individual species. When we drill down to that specific organism, we use a two-name system established way back in 1735 called binomial nomenclature. The genus is always capitalized, and the species is lowercase. And this brilliantly illustrates how these scientific names aren't just random, boring Latin. They're actually highly descriptive. Take Staphylococcus aureus, for example, a common bacterium found on human skin. Staphylo tells us the bacteria arrange themselves in grape-like clusters. Coccus means they're spherical in shape, and aureus comes from the atomic symbol for gold, meaning when these grow in a lab, they form golden yellow colonies. The name literally paints a picture of a cluster of golden spheres. How cool is that? If we jump back to the very top of that hierarchy, the domain level, our current system was actually proposed in 1978 by a guy named Carl Woese. He didn't just group things by how they looked, he compared the genetic sequences of their ribosomal RNA. The more similarities in the genetic code, the closer they were related. This massive breakthrough gave us the three-domain system. we have domain bacteria, which have cell walls made of a unique protein sugar mix called peptidoglycan. Then domain archaea, which lack peptidoglycan and love those extreme environments. And finally, domain eukarya, which houses everything with a true nucleus. Get this, though. Genetically speaking, those extreme archaea are actually more closely related to us in the eukarya domain than they are to bacteria.
Section 3. Shaping our planet. It's time to look at the global impact of the unseen. We humans tend to have a pretty human-centric, or at least a plant and animal-centric view of the world. If I asked you right now what produces the oxygen we breathe, you'd probably say trees and plants, right? But the microscopic reality completely flips that perspective. Photosynthetic microorganisms, like cyanobacteria and algae, are actually responsible for roughly 70% of the Earth's photosynthesis. Microbes, not plants, are the true lungs of our planet. They literally drive the structure and content of our soil, our water, and our atmosphere. To really put their dominance into perspective, just think about this number: 3.8 billion. The Earth itself is around 4.6 billion years old, give or take. The very first single-celled living organism is believed to have shown up roughly 3.8 billion years ago. That means for billions of years, long before the very first plant or animal ever existed, microbes were busy shaping the Earth habitats, cycling gases, and basically laying the groundwork for all the complex life that followed.
Section 4. Solving microbial mysteries. Let's look at the epic showdown between spontaneous generation and biogenesis.
For centuries, there was this massive, intense scientific debate about where life actually comes from. Many people believed in spontaneous generation, the idea that some invisible vital force in the air could just spontaneously create life from non-living matter. Others argued for biogenesis, meaning life only comes from pre-existing life. In 1668, Francesco Redi proved that maggots didn't spontaneously appear on meat if you just covered it so flies couldn't land. Then in 1765, Spallanzani showed that boiled broth in a sealed flask didn't grow microbes. But critics pushed back, claiming that sealing out the air destroyed that magical vital force. Finally, in 1861, Louis Pasteur designed this brilliant S-shaped flask. He boiled the broth, but left the flask unsealed so air could freely enter. However, the S-curve trapped all the dust and microbes in the neck. The broth stayed perfectly sterile. Boom. Pasteur definitively proved biogenesis. Air alone does not create microbes.
But honestly, none of those debates could have even happened without the pioneers who actually saw these organisms first. Back in the 1670s, Anton von Leeuwenhoek was squinting through these primitive, handmade microscopes at rainwater and scrapings from human teeth, of all things. He was the very first person to describe living microorganisms. He enthusiastically called them animalcules, or little animals. He sketched rods, spheres, and a massive diversity of life that no human being had ever laid eyes on before. Following Pasteur's breakthrough, we entered the golden age of microbiology. It was just a rapid-fire era of discovery. First, Ignaz Semmelweis tragically had to fight the medical establishment to prove that doctors simply washing their hands with chlorinated lime between autopsies and delivering babies would drastically drop mortality rates. Then, Joseph Lister applied this germ theory to surgery, using carbolic acid to pioneer aseptic surgical techniques. And finally, Robert Koch developed a strict set of logical steps known as Koch's postulates, involving isolating microbes from dead mice and injecting them into healthy ones to definitively prove that specific microbes cause specific diseases. The story of Alexander Fleming is one of my favorites, and it brilliantly illustrates how science isn't always perfectly planned. In 1928, Fleming was trying to grow a plate of Staphylococcus aureus bacteria. He left the plate out and it accidentally got contaminated with this fuzzy mold. But Fleming made a genius observation. Right around that fuzzy mold, the bacteria just couldn't grow. The mold was secreting something that inhibited the bacteria. That mold was penicillium, and Fleming had just accidentally discovered penicillin, the world's very first antibiotic. It was a total lucky accident that completely revolutionized human medicine forever. Section 5. The Modern Microbial Revolution. Let's see how we use biotechnology and explore our very own microbiome. In the modern era, we've kind of shifted gears. We stopped just fighting microbes and actually started harnessing them. In the 1970s, scientists discovered restriction enzymes in bacteria, which are basically molecular scissors. This allowed for recombinant DNA technology.
We can literally take the human gene for insulin, splice it right into a bacterial cell, and have that bacteria pump out pure human insulin for diabetics. It's a drug called Humulin. And then in the 1980s, we developed PCR, which acts like a DNA Xerox machine. It lets us take a tiny microscopic trace of genetic material, like from a crime scene or a viral swab, and amplify it into massive quantities so we can easily study and identify it.
The absolute most crucial thing we are learning today is that humans are essentially walking ecosystems. We've mapped the human microbiome and discovered that the vast majority of microbes on and inside our bodies are completely harmless or even highly beneficial. They aid our digestion and regulate our immune system. We're finding that babies born via C-section acquire a very different baseline of microbes compared to babies born vaginally, and that impacts their early health. We are even treating severe antibiotic-resistant gut infections like C. diff using fecal transplants. Yeah, literally introducing feces from a healthy person into a sick patient's gut so the good bacteria can outcompete the bad ones. It sounds wild, but it's a real game changer.
Which brings us right back to our original question. Are you ready to rethink your relationship with your own microbiome? If our health entirely depends on maintaining a diverse, balanced ecosystem of good bacteria on our skin and in our guts, perhaps completely sterilizing our environment with harsh antibacterial chemicals isn't the best idea after all. We are not just individuals. We are vast, microscopic communities. Thank you so much for joining me for this explainer. Keep wondering, keep learning, and I'll catch you next time.